Hi, everyone. My name is Emily, as Joe said, and I'm one of the MindCORE postdoc fellows. I'm really excited to get to talk to you guys today. Joe said the new fellows. I've been here a year, so I'm like new-ish, but always getting the lay of the land. So I'm going to talk to you all about my research on the development of conduct problems through a biopsychosocial lens. Okay, so as Joe said, I'm a clinical psychologist by training. My current work draws on basic science approaches to understanding mental illness, while also keeping a firm grounding in the realities of the clinical conditions that I study. Throughout my PhD, I balanced conducting neuroscientific and cognitive research into behavioral and emotional dysregulation with working directly with children, adolescents, and adults who are experiencing these kinds of problems. Including those whose impulsive or callous behavior had landed them in legal trouble. So I worked in the juvenile justice system. These experiences create kind of a feedback loop for a lot of clinical scientists, in which I applied scientific knowledge in assessing and treating clients, and also identified research needs and generated hypotheses to test based on what I observed in the clinic. So the questions that have captured my interest as a clinical researcher are about the development of conduct problems, the kinds of behaviors that embroil people in the legal system, things like aggression and serious rule violations. My research focuses on how we can identify and help youth who are starting to show a maladaptive trajectory of behavior problems. So how can we divert their paths by fostering resilience and ultimately by changing the systems that put them on these paths? So to answer these questions, I've applied perspectives and techniques from multiple fields, including clinical psychology, developmental science, public health, criminology, and psychophysiology. And through this integration of disciplines, my work directly contributes to novel approaches to assessment and to intervention, which can be applied to both individual treatment approaches and also policy reforms. So broadly, I integrate biological, psychological, and sociocultural levels of analysis to conceptualize conduct problems and develop solutions to decrease risk and foster resilience. And in the time we have today, I'd like to walk you through some work that I've done in each of these domains. Am I talking loud enough? Can you guys hear me? I can't tell if the mic is on. Okay, awesome. So let's start with a couple of recent studies coming from a psychological perspective. These are focused on dispositional traits that confer vulnerability to conduct problems. So this line of my program of research has concerned personality and its antecedents and how we can use these emerging traits to shed light on risk for chronic conduct problems over adolescence and into adulthood. And in particular, I'm interested in individual differences in callousness, which we can think of as a dispositional lack of social emotional sensitivity, as well as disinhibition, which is an impulsive, unrestrained cognitive behavioral style. And you could see how these different traits might intersect to create a kid who is relatively well-controlled, but sort of cold and calculating, one who is dysregulated and impulsive without caring about the effects it might have on other people, another who has big dysregulated emotions and behaviors, but who has affiliative bonds with other people, and finally, one who is both relatively well-regulated and pro-socially oriented. And these traits have been described, albeit using a variety of terminology in the clinical, neuroscience, developmental, and criminology literatures. So the first study I'll talk about, we use the multinational Imogen study, which is over 1,000 European community adolescents, to explore how questionnaire measures of callousness and disinhibition at age 14 could serve as markers of risk for the escalation of conduct problems over 2 years. So we compared findings for conduct problems specifically with other kinds of dysregulated behavior often seen among teenagers, including ADHD-related impairment and alcohol use. And remember, this is Europe, so they start a little bit earlier. All of our analyses controlled for any symptoms that the kid was already showing at baseline, as well as demographics like age, gender, country. So here's what we found. On the y-axis is the incident rate ratio for each predictor. And you could see that trait disinhibition, which is the black bars, at age 14 was uniquely associated with the escalation of all kinds of dysregulated behavior by age 16. So this trait can be considered an indicator of risk for what we call externalizing problems, broadly speaking, these dysregulated behaviors. And callousness, on the other hand, on the gray bars, shows a selective association with the escalation of conduct problems and not with other types of dysregulated behavior. So this suggests that the trait operates more narrowly, guiding the manifestation of conduct problems in particular. And given the nature of callousness, an implication is that atypical patterns of social and emotional processing may contribute to engagement in behaviors that hurt other people or are considered, generally condemned by society. And this is consistent with models of psychopathic personality, in which callousness is typically a central feature. So a large part of the reason why I decided to come to Penn as a postdoc was to get the chance to explore how these teens became callous in the first place. I knew from my research that both genetic and environmental influences were important, and that this was really clearly illustrated by my clinical work with teens who were facing serious felony charges. From talking with them and their families and from reviewing their records from school and court, it was really clear that there was some component of temperament here, some early emerging style of relating to other people and the world that are observable from infancy, as well as interactions with parents and with broader systems in their world, like neighborhood, police, schools, things like that. So I wanted to understand how all of these factors combined to shape the kids that I met in juvenile detention, who seemed so hardened to the world. The most prominent model of how little kids become callous adolescents was described by Penn's own Becky Waller, together with Nick Wagner at BU. So they propose that there are two dimensions of temperament that are key developmental antecedents of callousness. One dimension is the degree to which affiliative social interactions, like cuddling, are perceived as rewarding versus neutral or uninteresting. The other dimension is threat sensitivity, which ranges from relative fearlessness to extreme negative reactivity to novel and threatening stimuli. So here you can think of a little kid who isn't afraid to explore new things and take risks, but also has a positive and loving relationship with a caregiver, one who is very reactive to perceived threat and seeks comfort from the caregiver, one who's quite fearful but isolates rather than seeking out a caregiver when scared, and one who isn't afraid to take risks, but also doesn't really have a strong bond with his caregiver. And the model proposes that it's this last child who will start to develop a cold and callous interpersonal style, especially in environments where their parents are overtly harsh or low in warmth. So we tested this model in a study led by another Penn psychology postdoc, Yael Paz, using data from the Adolescent Brain Cognitive Development Study, which is a huge longitudinal study at several sites around the US. And specifically, we tested the interaction between these two dimensions of temperament, threat sensitivity and affiliative reward, in predicting callous and emotional traits. Callous and emotional traits are a specific manifestation of conduct problems that involve predatory aggression and disregard for others' well-being. And to demonstrate the specificity of these temperament dimensions to callous and emotional traits, we also tested whether any interaction effect would be observed for a more general measure of conduct problems. So here are the results plotting that interaction effect for callous and emotional traits. On both graphs, you could see that threat sensitivity is on the x-axis with separate lines for different degrees of affiliative reward. The y-axis in the left graph is CU traits, callous and emotional traits at ages 10 to 11. And on the right is callous and emotional traits at ages 11 to 12. And in both cases, the highest CU traits at the top left corner of the graphs are observed for kids who are showing a combination of low threat sensitivity, like fearlessness, and low affiliation, so relative social disinterest. And this is controlling for conduct problems more generally, meaning that the outcome here is specifically the callous tendency that these kids are starting to show, regardless of their overall behavior problems. And this interaction effect was specific to callous and emotional traits and was not observed for conduct problems more broadly. Okay, so we've discussed how questionnaire measures can index traits that promote risk for conduct problems. And I want to move now to discussing biological and specifically psychophysiological approaches to capturing risk for conduct problems and their potential applications in real-world settings like the psychology clinic and the courtroom. So the work that I've mentioned so far has relied on these gold standard clinical psychology measurement approaches, questionnaires, clinical interviews. These are really important ways to understand the development of conduct problems because they can shed light on the individual's psychological experience, their typical behavior, right? These are the cornerstone of psychology. But they also have limitations, especially when we're working with people who have severe emotional and behavioral dysregulation. The usefulness of these measures depends in part on insight, that is, knowing why you act or think the way you do. And report-based measures also rely on what people are willing to tell you. And that might be limited in situations where those behaviors are stigmatized or criminalized, or when there are compelling reasons to present yourself in a certain way. So for example, I worked with kids who were facing serious felony charges who knew that they should say that they felt remorse for their behavior, even if they didn't, just so that the judge wouldn't think that they were cold-hearted. On the other side of the coin, we also know that the same kids might be motivated to act kind of cold and hard around their peers to fit in. And finally, knowledge of current symptoms, while helpful, doesn't actually tell us that much about the mechanisms and the processes that led to those clinical problems. And this is especially an issue for research on conduct problems because we know that there are a variety of pathways that can lead to the same types of behaviors. So for all these reasons, it's really crucial not to rely too much on any one source of data when we're studying risk for conduct problems. There are a lot of different ways that we can measure the same underlying risk-promoting trait. So we'll talk about some of my research exploring the physiological markers of the risk factors that we've talked about. So let's get into some of this research to understand the neuroscience of liability for conduct problems and other forms of dysregulated behavior using electroencephalography or EEG. As many of you know, this is a way to quantify brain activity using electrical potentials that make it through the skull and can be measured using electrodes on the scalp. I use EEG to look at brain responses to visual, auditory, and tactile stimuli occurring on the order of milliseconds after the stimulus is presented. And this approach of looking at an average response to a certain stimulus over the course of several trials is called the event-related potential technique, or ERP. So we're going to move from talking about the threat sensitivity and affiliative reward traits to talking more about general dysregulated behavior, more akin to disinhibition in its degree of generality across different kinds. So in this work, one of the ERPs that I study is called the P300, which is depicted here. On the x-axis is time in milliseconds, with 0 being when the stimulus was presented. And on the y-axis is the amplitude of the brain response in microvolts. On this head map, you can see that the response is largest, darkest red, over parietal occipital regions. So the P300 is largest following an infrequently presented but task-relevant stimulus. And in this case, one type of picture is presented over and over to the participant. It doesn't require a response. That's the blue line there, the standard. But once in a while, a different picture comes up and the participant has to categorize it by clicking a button. So the rare target stimulus that they're looking for elicits a brain response shown in the red line. And this cognitive response is thought to reflect the initial appraisal of the stimulus's significance, recognizing this is the special picture that requires a response based on the current rule set. Because P300 reflects in part the allocation of goal-directed top-down attention to categorize the stimulus as task relevant, it's seen as relevant to dysregulated behaviors involving unsuccessful deployment of executive functions. And indeed, we know that adults who are exhibiting more externalizing problems or dysregulated behaviors show a smaller P300 response. You can see on this graph, in looking at the top line there, that one, that adults with the lowest externalizing scores have the largest P300 response, whereas those with higher externalizing have a relatively blunted P300. And you could see as the externalizing score gets more and more extreme to the highest 10% of externalizing scores here, the response gets more and more blunted. Other research that I don't have time to go into right now suggests that the association between externalizing and P300 is genetically influenced, based on twin studies, as well as studies that look at having a relative who has severe dysregulation, like looking at the kids of people with substance use disorders, for example, as well as people who are not currently showing severe dysregulation, but who will go on to show severe dysregulation. So all of these forms of studies together tell us that there is some genetic component to the relationship between P300 and externalizing. So this gives us some clue that P300 is not just a neural correlate of existing problems where we don't know, what came first. It may serve as an index of the underlying genetic risk for behavioral dysregulation. And we might be able to use this marker as a way to identify people at risk before they start showing serious problems. So in a recent study, we tested this question using data from the Minnesota twin family study, looking at P300 at age 14 and its association with the escalation of behavioral dysregulation by age 17. And based on earlier research, we hypothesized that P300 would operate across different manifestations of behavior dysregulation, including conduct problems and problematic substance use, and would not be specific to any one kind of dysregulation, similar to disinhibition operating broadly, like we discussed before. We also examined the specificity of blunted P300 to behavioral dysregulation versus depressive symptoms, which are more indicative of emotional dysfunction. So here's the factor model of psychopathology that we used. In looking at the association between P300 and this general externalizing factor, we found that blunted P300 at age 14 predicted severe behavior dysregulation 3 years later, even once we accounted for the childhood manifestations of dysregulation like ADHD and oppositionality. So P300 was selectively associated with that common factor, explaining variance across all these different forms of dysregulation at age 17, rather than any one particular behavior problem. It was also not related to depression, supporting the idea that blunted P300 is a relatively specific index of risk for behavior dysregulation. So I've talked here about P300 as operating broadly across forms of externalizing, but we are also interested in applying ERP and other physiological approaches to understanding the more specific traits that I talked about earlier, including callousness, threat sensitivity, and affiliative reward. So the study that I just described is one example of a series of completed and planned studies examining the potential of incorporating biological measures into clinical assessment. Our study showed that P300 was picking up on some element of risk for serious problems later on that couldn't be forecasted just by looking at the existing symptoms the kid was already showing. In another paper that I collaborated on with Alex Callan at Florida State, our results suggested that including P300 in our assessments increased the precision of our predictions based on existing symptoms. So differentiating individuals who were showing relatively transient dysregulation from those who were at risk for a longer term course of problems. However, even though it's exciting to think about how we could revolutionize assessment practices by adding in brain measures, there are plenty of reasons to be kind of cynical or wary about it. First, with the current setup of the mental health care system in this country, it is impractical to get imaging done the same way you would at a medical appointment. I don't know of any community mental health clinics that have the time or space or even interest in adding EEG into their intake procedures on their own. And it would be really expensive and kind of unwelcome for researchers to try and distribute EEG systems to these clinics and organizations. Also, given the small effect sizes that we typically see for these types of models, with standardized coefficients hovering around like 0.1 to 0.2, the benefit of adding neural measures is heavily outweighed by the costs. But it's not all doom and gloom. In a recent commentary with my collaborator, Keenan Joyner at Berkeley, we laid out an iterative process for taking the questionnaire measures that we do commonly use in clinical assessment and refining them through reference to neural measures. to bring the measures we actually are able to disseminate into in the clinic into better alignment with our understanding of the neurobiological processes that contribute to mental illness. And this commentary that I've highlighted here just came out a couple of days ago, so I'd encourage you to go check it out if you're interested in reading further. Another context in which this kind of multimodal research could be applied is the courtroom. Recently, there have been some psychophysiologists and legal scholars who have become very excited about the potential to incorporate brain measures into criminal cases, whether as a way to show diminished capacity in some way or as a component of violence risk assessment. One of the things that gets people excited about this, and reasonably so, is the potential to reduce reliance on other metrics that might be more subject to racial and ethnic biases. So adding in neural measures theoretically has the potential to reduce the extreme racial and ethnic disparities that we see in the criminal legal system. In a recent paper for a policymaker facing journal, I collaborated with Roy Hamilton over in neurology, as well as others from across the country, to discuss some of the pitfalls of this approach. And I'm including the title here also because this also just came out in the last week, if anyone wants to take a more in-depth look. So we discussed the potential for neuroscience evidence to hold undue sway for legal decision makers, including judges and juries, because they may consider biological evidence to be more objective than other measures, and assuming that it's free of socio-political context, when this obscures the potential for significant bias in our neural measures and any science that we do. We also talk about how the complexity of neuroscience data and processing makes it very difficult for non-neuroscientists, including legal decision makers, to critically evaluate its merit. When these stakeholders need to rely on public-facing conclusions made by neuroscientist gatekeepers, all the biases and subjectivities that go into processing neuroscience data are rendered invisible. And finally, commonly used neuroscience techniques such as EEG and to a lesser extent fMRI have built-in phenotypic biases against hair types and styles that are socially coded as Black. And this leads to interpretability issues for a given defendant's brain data, as well as more broadly, a lack of appropriate comparison sample with of participants with similar demographic backgrounds. In short, as with the clinical applications, I'm skeptical that these uses of brain data in the courtroom will actually provide a benefit, particularly as regards decreasing racial and ethnic disparities in the legal system. And thus, it might not be something that we should actually be aspiring to for a variety of reasons. However, I will say that basic science research is cited all of the time in courtrooms. I have cited it myself, even without the defendant's own scans being introduced. So if we can improve representativeness in neuroscience research samples more broadly and make that information more generalizable, I think this kind of framework knowledge of how the brain works in general is an excellent way to translate our work into applied legal settings. So finally, I want to spend the last few minutes talking about the final part of the biopsychosocial model, the sociocultural approaches. This area of research is quite new to me, so I'll briefly discuss some ongoing work aimed at changing systems through policy reform and public health interventions. And all of this is through a really lovely ongoing collaboration with Naomi Goldstein right there, and the Juvenile Justice Research and Reform Lab over at Drexel. So the first project I want to mention here is an evaluation that Naomi's lab is conducting of a training program that all police academy recruits in the state of Pennsylvania are required to complete. Given the prevalence of negative interactions between police and youth, this training is designed to educate law enforcement on developmentally appropriate and trauma-informed ways to interact with youth, as well as to stimulate formal and informal conversation between the police and young people, especially youth of color from the community, to reduce stereotyping, improve intergroup relationships, and ultimately reduce racial and ethnic biases or disparities in youth arrest rates. Our analysis is focused on about 1,300 police recruits from here in Philadelphia who completed a survey before and after the training regarding their attitudes, beliefs, and behavioral intentions related to perceptions of and interactions with youth. So factor analysis of the surveys found 3 factors. First, calm and respectful intention. So items like, I try to keep another officer calm if I see him or her getting worked up. A second factor is police responsibility for positive youth interactions. So having positive interactions with young people in the community is an important part of my job. And third, sensitivity to the youth perspective. So things like police often overreact when dealing with young people. We also looked at a specific item about acknowledgement that race and ethnicity affects police-youth relations in their community. So in multi-level modeling, scores on all three factors increased from pre to post training, which is great, as did scores on this single item about race relations. And we saw the largest effect for sensitivity to the youth's perspective, suggesting that this face-to-face direct interaction with youth component of the training program may have been particularly helpful. And importantly, the training effects did not vary by any demographic characteristics, suggesting that this is similarly having an effect across a variety of different groups of police officers. So I was really excited to get to contribute to this study because so much of our work in psychology, and I think in a lot of the other areas that MindCORE focuses on, is focused on the individual, right? Like identifying the personal characteristics that contribute to and maintain mental health problems, in my case. And this study tests a systems level change, the implementation of a police officer training that could have downstream effects on the environments that kids at risk for severe conduct problems are regularly navigating. So by decreasing unnecessarily negative interactions with police in their communities, these kids are less likely to get embroiled in the juvenile legal system. So I think there's a lot of potential for this kind of approach. Another program that I've been helping the Drexel Lab with is the Positive School Safety Program, which they developed. Like the policing program, this program aims to change systems that put kids at increased risk for legal involvement and escalated behavior problems. And in this case, we know that a lot of referrals to the juvenile legal system stem from incidents occurring at school. Because dysregulated behaviors tend to escalate when a kid is surrounded by their peers and by adults who don't really know how to respond to the situation appropriately. And this is known as the school to prison pipeline. In this program, school staff receive training about child and adolescent development and trauma, and they learn skills to increase positive interactions with the students and decrease challenging behaviors without escalating the situation. The skills are cemented through a peer coaching model. And currently the program is running with two separate populations. One is school safety officers, like, law enforcement who are placed in schools, and school climate staff. So that would be like cafeteria monitors, people who provide supervision during unstructured times of the day at school. So I think this program really beautifully illustrates how changes to systems have the potential to filter down and influence individual youth's behavior and mental health. And in this case, hopefully reducing the escalation of conduct problems to the point of arrest. So I hope I have adequately conveyed my enthusiasm for the different approaches that we can take to understanding the development of conduct problems. And I really want to thank my collaborators at Penn and elsewhere, and MindCORE in particular, for the fellowship, the support, and you all for listening. So thank you very much. We have time for questions. I don't know where you're going. No. Yeah, Sarah. Yeah, that's a good question. So for those who couldn't hear, the question was about like why P300 is, is that expected to be related specifically to impulsive behavior? And the answer to that is that over decades, like there's this huge long research tradition on looking at substance use disorders in particular and P300. It's an excellent question that I will have to look up later about like, why did they start looking at that in the first place? But drawing on that, the evolution has sort of been like, okay, this is about substance use disorders. Wait, but it's also true about like kids of parents with substance use disorders and people who like go on to have substance use disorders. Okay, but now we can expand it to other kinds of externalizing. And so it's been this really iterative process. But yeah, I'm not actually sure why they decided on the P300 to begin with, but from sort of a post hoc explanation perspective, the P300 is really closely tied in with executive functions. And we think that that is at the core of what makes externalizing problems cohere together.
Thank you.
Yeah, thank you. Yeah. Thanks for coming.
Questions. The first one, when you see a small effect size, isn't it because like a small population? And is it possible to kind of like, because I know there's previous research at like 23andMe, and there were people who are in a genotype, they're able to like see like, oh, certain like this genetic basis of being able to taste cilantro differently. So could you have kind of like a corporate partner and kind of people who've already been genotyped and do these kind of studies? Because for like, maybe not the P300, but if you want to look at genetic basis, then you can already have people's genotypes. Like, would that be an effective way to kind of further this investigation?
That's a great question. And there are a lot of people who do genomic work on conduct problems. A couple of things that I'll say about that is that the small effect sizes that we're seeing are not because the sample is too small. And in fact, you often see larger effect sizes with smaller samples. You get more of a precise and often really small effect size when you get to a larger sample, including, you know, tens of thousands of people in it. I would also say that the effect sizes that you see for things like, you know, cilantro tasting and whatnot are a little bit simpler in terms of genetic influences that, you know, that drive that experience, that perceptual difference. Whereas these behaviors are so many sort of units of analysis away, so many different like levels away from coding particular proteins with specific genes that often they require this huge number of different genes that we're looking at. And still, even when you look at across the entire genome, the effects are still pretty small, which just suggests that there's a lot of things as serving as intermediaries in the pipeline between, you know, here are, you know, my chromosomes and my genes and whatnot, and the eventual behavior that we see in the real world. Does that answer your question? Okay, thank you. Awesome. Thank you guys so much.